All right, so now we're going to discuss how cancers develop on a molecular level. So we already talked about how DNA can be altered, and if DNA is altered, you can develop mutations and then run the risk of cancer, right? So we talked about that. But now we're going to talk about, on a molecular level, how cancers develop. And they really can develop by one of two ways. So, one, you can increase the proliferation. That's one way. Just increase the division of cells. Have uncontrolled division. Or you can decrease the suppression. All right? So those are the two main ways. You basically think of it like driving a car. So put your foot on the gas and speed it up more, or take your foot off the brake. Those are the two ways that you can lead to cancer. Let's first talk about how cancers arise from increased proliferation, okay? That's what we'll talk about first. And we're going to discuss oncogenes in regards to this specifically. So oncogenes encode for normal protein products that are essential for cell growth and differentiation. However, if certain mutations arise, these oncogenes may actually become unregulated and lead to increased cell growth and potentially neoplasia. So categories of oncogenes include growth factors, growth factor receptors, like we see here, signal transducers, so downstream effects, nuclear regulators, so in the nucleus with the DNA, all right, and cell cycle regulators, so transcription factors and the more downstream proteins. So let's now discuss all these things in detail and each of these steps involved in oncogenes. All right, so without further ado, let's begin. And let's start off by talking about growth factors. So the things that bind to the receptor right here. So growth factors. Well, they induce cellular growth, thus increase activity or number of cell proliferations, and therefore they will increase growth. So, can you think of a specific growth factor oncogene known for increasing, say, astrocytomas or brain astrocyte tumors? Well, you should probably know that it is platelet-derived growth factor. Okay? The same thing involved in wound repair. That thing, if it's increased too much, it can actually lead to astrocytomas. Not very good. So that is associated with growth factors. So normally, again, what we're going to do is describe the normal steps involved in growth and then talk about the abnormal with it. So growth factors normally transmit signals from the outside environment to the cell. And the more receptors that there are, the more signals that can be communicated to the cell to tell that cell to grow and divide. So let's now talk about the receptor, okay? The receptor right here. What receptor is associated with a decreased prognosis of certain breast cancers, and what medication specifically inhibits it and treats it? So that would be the cancer gene ERB, ERB, ERB2, or another name is HER2/neu. And what is the drug that actually treats it? Trastuzumab, or Herceptin, is a monoclonal antibody that actually binds to these increased receptors and inhibits them. So that's important to know, and that's associated with a decreased prognosis, actually, in breast cancers. So we talked about the growth factor and the growth factor receptor. Now, once the growth factor is bound to the receptor, a tyrosine kinase that's attached to this receptor on the inside, and kinase means what, again? It means phosphorylation. So something that phosphorylates the receptor, this can also be increased in activity and lead to cancers if it's too much, if the activity is too much so. So what type of mutation is known to cause an increased tyrosine kinase activity, and specifically in the cancer chronic myelogenous leukemia, CML? And what chromosomes are involved? This is a very important question. You really need to know this. So the genes involved are the ABL gene, ABL gene from chromosome, do you know? From chromosome 9. And it involves a translocation. So a fragment of this is stuck onto chromosome, what chromosome? 22. So chromosomes 9 and 22 are switched up, are translocated. And what is the gene on chromosome 22? BCR. So this chromosome that has a piece of 22 and 9 on it is called what? There's a special name for it. It's the Philadelphia chromosome. You need to know that. It involves the translocation of 9 and 22 in the genes BCR and ABL. And this leads to constitutive phosphorylation of receptors without the need for growth factors or growth factor receptors. So this tyrosine kinase is just constantly going and going and going and telling the downstream effects to just keep going and going and going and proliferating over and over again. Very, very common, commonly tested on the U.S. Assembly. So there's also another monoclonal antibody that can be used to bind to these tyrosine kinase receptors that are overactive. What is that antibody? What is the name of that medication? It's imatinib. Another word is Gleevec, another name, the trade name. Very good. So imatinib. Okay, so once we have phosphorylation on the inside, this actually leads to stimulation of RAS proteins that replaces associated GDP with GTP. So normally you have GDP. Well, now that's stimulated, we have GTP now. So this GTP will then go and activate other things downstream. And before we go ahead with that, let's talk about when RAS proteins can be overactivated. What types of cancers do we see the overactivation of RAS? Colon cancer. Colon cancer, commonly mutated with the RAS gene. All right, so activated RAS and bound GDP then stimulates the MAP kinase cascade. Okay, that's pretty complicated, but you need to know all these steps. And this ultimately results in the synthesis of the transcription factor MYC. Okay, MYC. An increased activity of this in several different types of MYC, including c-MYC, n-MYC, and l-MYC, are associated with different types of cancer. So specifically what? What if there's overactivation of the c-MYC? What type of cancer is that? Burkitt's lymphoma, okay? What about the N? N stands for neuroblastoma. The L, lung cancer. So that makes sense. Except for the C stands for Burkitt's. All right. Well, with Burkitt's, you need to know the specific mutation. It's another translocation. There's 2 chromosomes that swap fragments. What are those chromosomes? So the c-MYC gene, what chromosome is that on? It's actually on chromosome 8. And that gets stuck onto chromosome, do you know? Chromosome 14. So what's on chromosome 14? Chromosome 14 actually houses the heavy chain. So every time that this cell wants to make a heavy chain fragment of an antibody, it's going to then make chromosome 8 as well, because that's stuck onto it. So it's going to make a MYC gene every time it makes a heavy chain. And as you know, if this is a B cell and it makes lots and lots of antibodies, then it's going to make lots and lots of MYC protein, and it's going to have a lot of proliferation. Got it? Good. So 814, Burkitt's lymphoma, MYC oncogene, know it. Good. All right. So let's then talk about the downstream effects of MYC. So MYC serves as a transcription factor for the synthesis of a protein called cyclin D. Now, what depends on cyclin for kinase activity? It's a very tricky question. How about what depends on cyclin for kinase? Cyclin-dependent kinase. That's it. Cyclin-dependent kinase depends on it. It's very, very hard to think about that one. So, CDK, or cyclin-dependent kinase, remember, what does the word kinase mean? It means phosphorylation. So, the CDK, once cyclin D is bound, is able to phosphorylate something. What does it phosphorylate? An inhibitory protein. It phosphorylates the RB suppressor protein. Remember we talked about RB. What cancer was this associated with? Osteogenic sarcoma, right? Right. Okay, so anyway, so cyclin-dependent kinase phosphorylates RB, and after this is phosphorylated, RB lets go of this E2F protein, and E2F is responsible for stimulating the cell cycle. In which phase of the cell cycle will this trigger the progression of? So here's the cell cycle, G1 to S to G2 to M. M stands for mitosis. What is the very important checkpoint phase of the cell cycle that you must know? It's the G1 to S phase. You have to know that's the checkpoint phase of the cell. It's because in the S phase, what happens? S stands for synthesis. S is where DNA is synthesized, okay? That's where you make DNA. So it's important that you have a checkpoint before you make more DNA. Makes sense, right? So this E2F protein is inhibited almost all the time so that you're not always progressing to the S phase. And the RB protein is responsible for that. But if the RB protein is phosphorylated because the cell is activated by this growth factor to start growing, then the RB protein will be inhibited and the E2F protein will be free to cause the progression from the G to the S phase. G1 to S, know that for sure it's going to be asked on your test. So RB protein is responsible for monitoring that progression. There's another protein too that we'll talk about in a little bit.